Okay, so I'm going to talk about a study in which we used partial least squares regression method to predict the mean bold activity in the fusiform phase area based on the pattern of activity from other visual cortical areas. So I'm actually going to talk about the study that we did, so it's not more like introducing a new method or something like that. Okay, so as you know, area FFA or fusiform phase area is one of the most prominent phase selective areas in the human brain, which responds selectively to images of faces compared to images from other object categories. However, it's not clear how cortical areas are contributing to the preferential response in FFA. And even though there are some fMRI studies showing specific functional connectivity patterns between FFA and other phase selective areas, however, the contribution of non-phase areas to the responses in FFA is less known. So here we decided to develop a framework based on the regression analysis to see how other visual cortical areas are contributing to the responses in FFA. So first of all, we defined some areas, some ROIs. So some of these areas are classic retinotopic areas that we define based on a probabilistic retinotopic map. So there is an atlas of retinotopic areas, which is based on the average of 53 subjects. So we used that atlas and projected this atlas onto the surface of individual subjects. And then we identified some of the well-known retinotopic areas in each subject, including area V1, V2, V3, some areas along intraparietal sulcus like IPS 0 to IPS 4, some areas in some mid-level areas in ventral occipitotemporal cortex like area HB4, VO1, VO2. So in total, we had about 16 retinotopic areas. We also identified some functional areas using functional localizers. So we had a motion localizer to identify area MT, which is a motion selective area. We also had localizers to identify face areas, body areas, and place areas. So in total, we have 24 areas, and we want to see how these areas are contributing to the responses in FFA. So in order to address this question, we ran an fMRI experiment in which we presented some naturalistic movie clips. So in each run, we presented 60 movie clips. And these clips were drawn from different action categories like human interactions, human object interactions. So the idea was to drive the visual cortex as much as we can using a variety of rich visual stimuli, and then see how the areas are interacting with each other. So a very quick overview of the method. So we had 10 runs in each subject. We scanned subjects in a 3D scanner. We analyzed data on the surface, and also we did a little bit of spatial smoothing. And we analyzed data separately for left and right hemisphere. Okay, so a simple way to look at the relationship of activity between the between two areas is to run a correlation analysis. So similar to what you do in resting state function connectivity analysis, you can take the mean of activity, for example, in FFA, and the mean of activity in another area, and then compute the Pearson correlation. So for, let's say, 60 stimuli, you get basically beta values for these 60 stimuli in FFA and in a given area, and then you compute the correlation. The other approach that we took was to come up with a regression model and then try to predict the mean activity in FFA based on the pattern of the response in other areas. So I'm going to describe this regression model, and then I want to explain why we use this particular regression, which is called partial least squares regression. So in the regression analysis, do we have pointer? Okay. So in the regression analysis, we have a training phase and a testing phase, of course. So in the training phase, you take 59 stimuli. So your dependent variable is a vector of 59 stimuli, and you have beta values here. Your independent variable is a matrix of 59 stimuli by the number of vertices in a given area, and then you estimate the weights for these vertices. So basically, we want to see how each individual vertex is contributing to the response in FFA. And then you take these weights to predict the mean response in FFA for the left out stimulus. So you give the beta values of the left out stimulus here, and then you multiply it by the weights, and you come up with the predicted response, and you repeat this procedure 60 times. You get a vector of predicted activity in FFA. You also have the actual values in FFA, and then you can measure how these two are correlated. So we used explain variance measure to basically compute the accuracy of the model in predicting the mean response in FFA. So this is a simple regression framework. So why we use partial least squares regression? The reason is that, as you know, one of the common problems in the regression analysis is the collinearity problem. So for example, here, you know that the vertices are not fully independent, so the activity in the vertices are highly correlated, especially in one area. So using this method, you basically try to find some components, like few components, and then you use those components as independent variables. So instead of using individual vertices, you basically use those few components in your model. So the idea is very similar to run a PCA, and then you work in a lower dimensional space. There are also some similar methods like support vector regression and L1, L2 norm regression. They also have this superiority over ordinary least squares regression, so they can overcome the collinearity problem. The other advantage that we saw in our pilot analysis was that this method has a better generalizability. So like when you go to predict a new stimulus, you usually get a better performance using partial least squares regression. So here we have the results for the 24 areas. So if we do the correlation analysis, many areas show good correlation of activity with FFA, which is not very surprising. So for example, some of the early visual areas have a good correlation. Some mid-level areas like HB4, LO1, LO2, the body areas, motion selective area, and also the other phase selective areas, they all have good correlation of activity with FFA. The areas along IPS don't have a good correlation, and also the place areas, they don't have a good correlation. So even though like one of the place areas, which is called parahippocampal place area, is located next to FFA, and it is sometimes assumed that the activity in these two areas are negatively correlated, the activity between FFA and PPA, but actually the place areas are not good. They don't have good correlation with FFA. So if we do the PLSR analysis, we get somewhat similar results, except that in early visual areas, we don't see a good prediction. Like areas like V1 and V2 do not have a good prediction power for predicting the mean activity of FFA if we use this regression analysis. So one question that may arise is that these correlations and also these regressions may be derived by stimulus-related correlations. So because we have shown a stimulus, some of these correlated activities between areas may be related to the stimulus that we have presented. So in order to address this question, so we went back to our GLM model, so the model that we use to extract the beta values for these 60 stimuli. So instead of using the beta values, we use the residuals of the GLM model. So the residuals of the GLM model are supposedly stimulus-free because we have explicitly modeled the stimulus component, so the residuals would be somewhat stimulus-free, so they would resemble the resting state signal. So if we use the residuals to do the correlation and regression analysis, what happens is that in many areas, especially in early visual areas, you see that the correlation increases. Like the correlation of the activity between V1 and FFA increases if you remove the stimulus. So it looks like the presence of the stimulus actually decorrelates the activity between FFA and many areas, and the reason is that the areas get engaged in the processing of the stimuli, and because the areas may have different functions, you would see less correlation of the activity. However, the areas that that seem to have strong interconnectivity with FFA, and so these areas include the body areas and the motion selective area. For these areas, the correlation actually decreases when you remove the stimulus. So actually, the presence of the stimulus increases the correlation. And we get similar result when we do the PLSR analysis. In general, the results of the correlation and regression analysis are correlated. So in these plots, one axis is the amount of correlations between FFA and other areas, and the other axis is the explained variance values that we get them from the regression analysis. The data points correspond to different subjects and different areas. So overall, the results of correlation and regression analysis are correlated. Finally, we can use this framework to predict the mean activity in another area, for example, in occipital face area, based on the pattern of activity in FFA. So like for 3 face selective areas, we can do all possible combinations, like take one area as a target area and try to predict its mean activity based on the pattern of the response in another area. And if you do that, you come up with a diagram like this. So the thickness of the arrow corresponds to prediction power. So when the accuracy of prediction is higher, and the arrow is actually thicker. So what you can see is that the mean activity in FFA can be better predicted from the activity in OFA. And also both FFA and OFA are good predictors of the activity for posterior STS face area. So if we assume that the area which is a good predictor of the activity is located upstream in the visual hierarchy, it's a very strong assumption, but if we assume that we have a scenario like this, we can use this kind of analysis to infer the hierarchical organization of the visual areas. Like for example, here we may conclude that OFA is located upstream in the visual hierarchy compared to FFA and PSTS, and perhaps provides input to these areas. So I can skip the conclusions except for one thing. So if we make a regression model and we include all the areas except FFA, and then try to predict the mean activity in FFA, so all non-FFA all non-FFA areas collectively, they can explain up to 75% of variance in the FFA response, which is a quite large amount of explanation power. Right now, one thing that we are doing is we are actually going to use the HCP movie data because in the HCP database, about 180 subjects have been presented with the movie stimulus. So we can, again, use this framework of analysis with this database. And instead of using, let's say, 24 ROIs, we can use a searchlight method. Like we can, for example, use FFA as a target area, and then we can have a searchlight ROI to predict the activity in FFA, and then we can move around this searchlight all over the surface and then come up with a map, like to see which parts of the brain can predict the activity in FFA well. And we want to use this regression framework for this kind of multivariate connectivity analysis. Yeah.